So hello everyone, welcome to today's Reverse Rett Bitesize session. We're really happy to be here and to see so many people joining us this morning. My name is Rachel Stevenson and I'm co-hosting today with Beth Johnson, who's from Reverse Rett as well. We are really looking forward to today's session, but first I just need to make a few housekeeping points. So just to say, first of all, if you can just flip your camera on and off quickly, dignity permitting. I know a lot of people are caring while they're also attending today, just so that we know everyone's who they say they are. We also ask that you keep your sound on mute until you want to ask a question, just so that the sound quality for the video is good. If you want to ask a question, you can put your hand up in the usual way, or you can use the little emoji thing that is the little hand held thing. Or you can enter it into the chat, and then either you can ask a question after the session, after Stuart's finished speaking, or if Beth will ask this question for you. If you happen to be watching on YouTube after the session, don't forget that you can also ask questions or give feedback by entering your questions and feedback into the comment box below the video. We really appreciate your input and participation as well. For anyone who's attending today, our guest needs no real introduction. So Dr. Stuart Cobb first published on Rett syndrome back in 2007 as co-author with Professor Adrian Bird on the seminal reversal paper. That paper led to a global awakening of interest in Rett syndrome amongst researchers. It was the first time people really looked at the treatable condition. It really was the first time that families were given any hope whatsoever that one day there could be treatment for Rett syndrome that would come from the research that could really make a significant difference to the lives of people with Rett syndrome. Stuart has over 20 years experience in the field of translational neuroscience. He's led academic research labs at University of Edinburgh and Glasgow. He is a Simons Fellow and Reader in Neuroscience at the University of Edinburgh, and is also now Chief Scientific Officer at Neurogene, a new company founded with a mission to bring life-changing medicines to patients affected by rare diseases. However, none of these accolades and achievements should put anyone off asking questions today. At Reverse Rett, we've been connected with Stuart for around 10 years now. And what we've appreciated most is not only his willingness to speak at events like these for us, but also his ability to ensure that the science is not too baffling, that it's explained in a way that's palatable and manageable so that everyone can engage. Stuart, we are really, truly grateful to you for taking the time out to do this for us today. And without further ado, I'd like to hand over to you.
Great. Thank you very much. Thanks for the kind invitation and introduction, Rachel. So I'll just put up my slide deck. And so as Rachel was saying, I'm based at the University of Edinburgh, and what I'm planning to do today is really to go over two things. So the first is really to give an introduction to the concept, if you like, of gene therapy. What is gene therapy and how is it applied in Rett syndrome? And then at the end of the discussion today, I'll give a sort of brief update on what's happening in industry and what things are looking like in terms of clinical trials downstream. So that's a general outline of the presentation today, but I'm going to start off really with the basics, and I'm very happy to take any questions you have either along the way or at the end. So I guess to start off at the very beginning, what is gene therapy? So gene therapy, and sometimes it's referred to as gene replacement therapy, is a really simple concept. And that is when you have a genetic disorder like Rett syndrome, where you have loss of function of a gene, gene therapy is really just putting back a working copy of that gene. So conceptually, it's really very simple. Now, we know that Rett syndrome is caused by mutations or faults in a gene called MECP2. And this MECP2 codes for a protein that is present in all cells in the body, but it's especially present in neurons in the brain. So the nerve cells in the brain have high levels of this MECP2 protein. And in Rett syndrome, you basically lack functional protein. So the concept of gene therapy in Rett syndrome is to basically replace the faulty gene with a working copy. So the concept's really quite straightforward. Now, one of the things we're often asked is, how do you actually go about making a replacement gene? And the answer to that is we synthesize the gene using sort of modern technology. So the old way, so basically a gene is basically a string of letters which code for making a protein. And so if you like, there are words, and these words code for protein, and it's a string of letters. And the MECP2 protein is around 1,500 letters long. So it's quite a long word, but basically there's a string of letters that code for making MECP2. That's all the gene is. It's just a string of letters. And what you can do is you can basically type out this long string of letters, and you can email that to a company, and they will take that string of letters in your email and they will convert it into that sequence of DNA coding those letters. So basically you type out the word, you email it to a company, and they use what's called a DNA synthesizer, and they will make a gene for you, and they'll send you a little tube with the MECP2 gene in a solution in that tube. So that's how it's done these days, and it's really quite a simple process. Now, there are different parts of the gene that go into making a gene therapy. So there's what's called the transgene, and this is the bit of the gene that makes the protein. So it codes for the protein, but there are other parts of the gene therapy which are things like switches and controls, which basically tell the cell when to switch on the gene and what level to produce protein at. So there are basically other elements. So these are called promoters and what's called untranslated regions. The technical details don't really matter, but basically you have a bit of the code that codes for the gene, and then other bits that basically regulate the gene. And this is what goes into a gene therapy. Now, once you've got the DNA that codes for MECP2 for the gene therapy, the big challenge is actually how do you deliver that as a drug? Because unlike conventional drugs, which are tiny small molecules, DNA is a very long, complex molecule, and you can't give it by traditional methods. So you can't put it in a tablet and take it as a tablet, because it would be destroyed in the stomach, and it wouldn't actually get to where you want it to go. So what's done with the gene therapy is you put the gene into what's called a vector, and a vector is really just a sort of capsule or formulation, if you like, that enables the gene to get into the body effectively. And there are two broad types of vector for gene therapy. One of them is called non-viral, and I'll not really talk about these because they're used much less. They're actually very inefficient for targeting disorders that affect the nervous system. And instead, I'm going to talk about viral gene therapy, because all of the companies that are working on gene therapy for Rett syndrome are developing what's called viral gene therapy. And all that means is that your DNA that codes for the MECP2 gene is packaged in a virus or a viral particle. And basically that viral particle is the drug product. That's what's delivered to the patient as a therapeutic. And the virus is really there to enable delivery of the DNA, the therapeutic DNA. And there's lots of advantages of using viruses. So people get concerned when you hear about viruses because they normally associate viruses with disease. And we've just gone through these 2 years of COVID, so everyone's very aware of how devastating viruses can be. But the key feature of these viruses is that they're what's called non-pathogenic. In other words, they don't cause disease. They're innocuous viruses, and they've been stripped of anything that causes disease. They're really just a vehicle to deliver the gene therapy. Now, the virus that pretty much all companies and projects to date for Rett syndrome gene therapy has been using is this virus that's called the Dino associated virus. And this is a tiny virus particle. So this is basically a sort of capsule that basically holds the DNA. And the reason it's used, this particular virus, is one, it's non-pathogenic, it doesn't cause disease. But the other reason is it's one of the few viruses that can actually deliver the DNA into the brain, which is the main target, the nervous system for treating Rett syndrome. And although this is a fairly new technology, it's not completely new, and there are actually licensed drugs which are based on AAV viruses, and the most well-known one is a drug called Zolgensma, which is a gene therapy that's used to treat a pediatric degenerative disease. And this drug has been now dosed into more than 1,000 patients. And there are other drugs that use AAV as a gene therapy as well that are beginning to appear on the market. So one of the challenges that's really faced industry is that unlike most drugs, which are small molecules and basically made by chemists, it's a chemical synthesis that you can actually manufacture in a chemistry facility. Gene therapies are what are called biologics. That means that they're actually made using biological processes, and they're actually much harder to make than conventional drugs, because what you have to do is you need to grow viruses in basically sort of cell factories. So basically there are incubators that grow up the virus in large amounts, enough to treat subjects, patients. But it's a fairly complicated process. So you grow the virus, and then there's a big purification step to make the virus really pure enough to be used as a drug. And that has really been actually one of the things that slowed down progress in gene therapy, because there were lots of technical hurdles. But again, industry is beginning to overcome many of these technical hurdles. So, for instance, when Zolgensma was first made, the clinical trial for Zolgensma was limited to a few patients. And the reason it was limited to a few patients is that's all the virus they could make. So even with their entire capacity, they could only make enough to treat a few patients. But there's been big steps forward in manufacturing gene therapy viruses now that will help with future programs. So the next question that's often asked is, how do you deliver gene therapy to treat a disorder like Rett syndrome? So as I sort of indicated, gene therapy is not going to be like a tablet that you take daily. It's going to be an intervention. It's going to be given by injection. And because the the primary target for the disease is the nervous system, there's sort of some unusual injection, what are called routes of administration, that are likely to be used. So the conventional way to give an injection is an intravenous injection. And the difficulty with that, though, is that the viruses don't get from the blood supply very easily into the brain. It's very inefficient because the virus, unlike a traditional drug, is a much larger molecule and it doesn't get into the brain very effectively by that route. So most of the gene therapy programs that are being developed by companies are being delivered directly to the nervous system, and there are different routes of administration. So one is through what's called lumbar intrathecal injection, which is an injection in the space at the bottom of the spine. So it's not into the spinal cord, but basically into the bottom of the spine. So it's a bit like an epidural injection for analgesia. And then other routes of administration are directly into the nervous system. And what they're injecting into is the blue here, which is the cerebrospinal fluid, which is the fluid that surrounds the brain tissue. So it's not directly into the brain tissue, but into the solution that bathes the brain. So that this is called cerebroventricular and cisterna magna routes of administration. So it's likely that the first programs will use these routes of administration. Now, another question that's often asked is, what will this therapy look like? Will this be a sort of one-off therapy, or will it be a sort of repeated therapy? I think for viral gene therapy, the answer is it's likely to be a sort of one-off therapy. It will be one intervention, and the effect of the gene therapy is likely to be very long-lasting. And this is because normally your genes sit inside the nucleus of the cell. So every cell has a complement of genes, which is contained within the chromosomes. And what you're doing with gene therapy is basically you're adding an extra mini chromosome, if you like, that contains a working copy of MECP2. And if you think of the nervous system, the neurons that you have as a child or the neurons you have as an adult, it's not like skin cells that are turning over. The cells in the brain are permanently there. So whenever you deliver this gene therapy, it's going to be long lasting because you're treating these cells that are there throughout life, and it will be a long lasting therapy. So there's not going to be redosing or topping up the therapy. It's likely to be a permanent intervention. And of course, that has advantages and disadvantages. So obviously anything that's very long lasting and enduring is quite very desirable, but it also has important safety implications as well, because unlike a conventional drug whereby if you get adverse effects you can withdraw treatment, with the gene therapy it's very difficult to go back. So you need to have good conviction that your gene therapy is safe. So with gene therapy, because many trials have now taken place for gene therapies for different rare disorders, we're aware of what some of the adverse effects are that you can expect, and the most prominent adverse effect is immune issues, because as you're aware, when you get a viral infection, your body mounts an immune response. So for everyone that's had their COVID booster, you might be aware that you feel a bit grotty the next day because your immune system's kicking in, and what that immune system's doing is trying to develop a response to the sort of artificial or foreign virus that's entered the body. And this happens with gene therapy as well. So you can get an immune response, and that's pretty common. And but because we know about that, there's normally interventions to counter that. So the immunosuppressant drugs are commonly given along with gene therapy, because you don't want to have severe symptoms of the immune response, but at the same time, you don't want the immune system to destroy the drug, the gene therapy. So it's getting that balance right that's quite important. Now, I've mentioned that the concept of gene therapy is quite simple, but actually there are some challenges with Rett syndrome, in that Rett syndrome is quite a complicated disorder. And one of the big complications with Rett syndrome is the individuals with Rett syndrome can vary quite a lot from one person to another. So in terms of the symptoms and what Rett syndrome looks like, and different people can look quite different. So there's quite a lot of variability, and so that's one thing, and it's explained, to a large part based on the genetics. But there is no conceptual reason why gene therapy shouldn't, have some effect or be effective in individuals, whatever the mutation and whatever the phenotype, if you like, or the symptoms of the disorder. There's no reason it should be more effective in one than the other, but it's a complication. But the other complication is that the level of MECP2 protein in the body, we know it's very important. So everyone with Rett syndrome has too little MECP2, and that's what's causing the symptoms of Rett syndrome. But equally, you get individuals with duplication syndrome and related syndromes who have too much MECP2, and that's a problem as well. So one of the big challenges for gene therapy for Rett syndrome, which is not the case for all genetic diseases, is that not only do you need to deliver the gene therapy, but you can't deliver too much because you don't want to push the levels too far in the other direction. So that's a big challenge that all researchers and companies working on Rett syndrome are aware of, and build that into their plans. Now, we've learned a lot from animal studies over many years, and I'll not go through all this, but the basic finding is that you can model Rett syndrome in animal models. So when you disrupt the gene and have low levels of MECP2, you get the same features as you see in Rett syndrome. So all of the symptoms, if you like, are become present in experimental animals, and particularly mice. And what we can show is with gene therapy, using viral gene therapy or using genetic tricks by switching the gene back on, we can see that we see improvement across a broad range of things. So we see improved breathing, we see improved use of limbs, what are called motor phenotypes. Motor function is improved, EEG is normalized to an extent, there's reduced seizures. So pretty much everything that's been looked at shows, at least in a mouse, some propensity that it can be corrected. What this looks like in patients with Rett syndrome, we need to see, because ultimately we'll not know until a clinical trial what the benefit looks like. But the animal data supports the concept of gene therapy for Rett syndrome. So I mentioned that the big challenges for Rett syndrome specifically is really this challenge that you need to get the level of MECP2 correct. And the reason this is difficult is because when you deliver a gene therapy, the virus goes everywhere, but it goes to some places more than others. So in other words, you get higher levels in some cells and some brain regions and some tissues in the body than others. So this is quite a challenge for gene therapy, and this is something that many of the companies and labs have been working towards. Okay, so where are we at the moment? So I'm sure you're all aware that there are two companies who had announced programs in Rett syndrome. The first one, Novartis, has been working on Rett syndrome for quite a number of years. So this work originated in a company called Avexis, which were then acquired by Novartis. So this gene therapy program actually had quite a long history. And unfortunately, at the end of last year, Novartis made an announcement that they were not pursuing their gene therapy program in Rett syndrome. And it's since emerged that the reason for that decision was that they had concerns that the effectiveness of the gene therapy was limited by the fact that the virus was not efficient enough in reaching the parts of the nervous system that it needs to reach. So what Novartis decided is that they would basically stop this, what was quite an advanced program, and go back to the drawing board, if you like, and try and develop a more efficient gene therapy and move forward with a more efficient gene therapy. So Novartis have not stopped working on Rett syndrome, but what they felt was that the program that they had acquired through Avexis was not as likely to be as effective as they would like it to be, and therefore they've gone back. So obviously that's a major disappointment, but that's a corporate decision that they have taken. The other company that's made the recent announcement is Taysha Gene Therapies. So this is a company that's based in Dallas in the United States, and they very recently, in last month, announced that they would be initiating a clinical trial for gene therapy, and that clinical trial is going to take place in Canada. So they got approval from the health sciences in Canada to conduct a phase 1 clinical trial for Rett syndrome, and that will be initiating this year. And essentially their gene therapy program is again a gene therapy program that's come out of an academic research lab and being taken into the clinic by Taysha. And Taysha are very good at promoting what they're doing, so I'm fairly sure that they will keep the community updated on how things are going there. So they're the two programs that people are aware of. There are also a number of academic programs. So we have one in Edinburgh that's quite advanced, and there's another one at the University of Pennsylvania, and there are other programs as well. And a number of these are already partnered with companies that are taking these forward into commercial development. What you find is that different companies have different strategies when it comes to what they present to the public. Some of them like to work away quietly, and others like to be very public. And it's just the companies just operate very differently. So there's these programs here that are quite advanced, and then there are various other companies working on different types of gene therapy. So I've only put up the companies here that are, have disclosed programs in Rett syndrome. So there's basically, these are slightly different flavors of gene therapy, but they're all genetic therapy, and they're all developing programs in Rett syndrome. But in fact, I guess what I would say is that there are many other companies that are working in Rett syndrome, developing genetic therapies. And it's not just the 4 here, it's very much in the double-digit number of companies that are now working in Rett syndrome. There's been quite a momentum really building up around this. I guess, you know, as we're talking about clinical trials and things, I guess it's fair to put in a slide on risks of gene therapy. So I guess the point I would make is that all first in human trials are safety trials. So that the goal of the trial is to show that the drug is safe. So that's the number one reason for the trial. But for gene therapy, there is an additional element, which is to look for an early readout of whether the therapy is effective or not. So that there are usually elements that will determine how effective the drug is, that's built into it, but they're primarily safety studies. And this is, you know, this is important to recognize. There is a difference between trials that occur in adults and children. So if it's a trial that occurs in a child, then so if it's a trial in an adult, then they only need to show that it's safe, and you'll get permission to go into a clinical trial. But if it's a trial that goes into a child, it needs to meet an extra hurdle, if you like, and it needs to be what's called show potential for direct benefit. And basically what that means is that it's got to have a likelihood of success in terms of being effective, not just safe. So gene therapies, you know, they have the potential to be curative, so they're targeting the underlying cause of the disease, but they do have unique safety issues. So one is it's an irreversible treatment. So once it's delivered, there's not really any going back with the viral gene therapy. We know about the immune responses. You know, this is very common, and there are interventions that are taken to mitigate against that. We know that specifically for Rett syndrome that the level of MEC2 is really important, and we don't want to overshoot there. And then, just like any other drug safety trial, there's the potential for unexpected effects that you don't really predict in advance. So there are risks, but of course there are potential benefits as well. And I guess one message I would make is that the risk of no treatment is very high as well, because we know that Rett syndrome is really, you know, devastating symptoms that if there was an option to improve, then one would want to do that. So there's risks of no treatment. So this is the balance here that the regulators consider before they allow a clinical trial to take place, but it's obviously the balance that you know, families take if they're considering you know, registering for a clinical trial. And I guess the last point on this slide is to make the point that whatever the therapeutic, the first generation of any drug is never going to be the best. There's always room for improvement. So the medicines we take now for whether it's you know, high cholesterol or heart disease or whatever, it's not the drugs that would have been prescribed 20 years ago or 30 years ago. Things get better, and therefore this first generation of gene therapy may not, you know, there's going to be room for improvement, but one needs to take a first step. I think this is my last slide, which is just to ask the question, you know, can we learn things from other disorders? And I would just flag up two here. So one, as I mentioned, SMA, spinal muscular atrophy, and this drug Zolgensma, which is you know, a licensed drug now in Europe and in North America and various other regions of the world. And this is a AAV gene therapy that is an effective therapeutic. So it's made it through onto the market and is effective. The other thing, this is a very different disorder to Rett syndrome, however, whereas this indication here, Angelman's syndrome, has somewhat more overlaps. It's obviously a bit different from Rett syndrome, but it has more overlap in terms of some of the symptoms. And there are 2 clinical trials on the go at the moment for genetic therapies for Angelman syndrome, and the results of the trials have not been published to date, but basically for one of the companies, they've released in a press release some sort of interim findings of the study, suggesting that they are actually seeing improvement in communication, behavior, sleep, reduced seizures, and also some improvements in motor control. So I guess this is encouraging. It's a different drug, it's a different indication, but there are some parallels with Rett syndrome, and I guess it's a chink of light that at least in another related indication that there seems to be some potential for benefit there. So I think that's it. I'll stop now, and I'd just like to acknowledge Reverse Rett and the sister organisation Rett Syndrome Research Trust's support over the years, and I'm happy to take any questions. Thank you very much. Thank you for listening.
Thank you, Stuart, for that absolutely brilliant presentation. And I feel like you really lived up to what I said at the beginning, which is that you have a way of making even the most complex information sound understandable. I would say learning that piece where you talked about the how this to the lab that synthesizes something I've always wondered about. And it was really interesting to hear you say how you have to type out the word, and hey, with MECP2, for example, there was
syndrome, you get one of the letters wrong and you lose the function of the protein. So yeah, so you check it's correct, and then when they send you back the DNA, then you need to read it again to make sure that they've not made a mistake at the other end. So that's really important. And actually, even when you make a drug product, when you make a Rett syndrome gene therapy, when you release the drug, you would probably still need to check that it's still right, even at that late stage, because the sequence is the most important thing.
So no space for typos. And
no.
This is a good segue to Beth taking over, because Beth does all of our proofreading at Reverse Rett. If you see anything from Reverse Rett that's spelt wrong, it's because Beth hasn't looked at it. So Beth, over to you to pick up the questions.
Thank you for that. No pressure. Yeah, I think just as Rachel said, I think actually as many people are watching, a lot of the questions that we might have had before you started, I think have been incredibly well covered. So we do end with some quite high level questions, as Rachel said. I'm happy to pose them, but if the people who ask them want to jump in at any point to fine tune whatever I've translated, or to then respond to what Stuart says, please do feel free to unmute and jump in. So Natalia has asked, will potential candidates need to be tested for their immune system response if such a thing exists?
Yeah, I think there's lots of different mechanisms of triggering the immune response, and it's inevitable that when you give an AAV virus, you trigger an immune response. I think I don't think there's an expectation that any given gene therapy will be more inducing of an immune response than in others. I think it's just a general feature of of virus-mediated gene therapy. What does happen in the clinical trials and with patients that are treated with gene therapy is that they're given, first of all, they're tested before they're dosed to see whether they have any existing antibodies against AAV virus, because some individuals actually do have existing antibodies. And then during treatment, that they're giving immunosuppressant drugs to dampen the immune system, and then that's tapered off. But then it's monitored so that there would be blood tests in the weeks and months after the gene therapy just to monitor the immune system. But there's not, for most people with Rett syndrome, it's unlikely that there's going to be immune response to the MECP2 that we're putting back. And the reason for that is that it's not a new protein that the body is seeing for the first time, which is normally the reason you trigger a strong immune response, because girls with Rett syndrome already have MECP2 protein in some of their cells, so that the body's familiar with it. And when you deliver extra, it's not seeing it as something new. So I think for Rett syndrome, unlike some other genetic diseases where the individual doesn't have any of that protein from birth, and therefore when it's suddenly presented as a gene therapy, there's a potential to mount an immune response to it. That will not be the case for Rett syndrome.
Natalia, does that answer your question?
Yes, that sounds good, actually. Yeah, that's a good point, because obviously it's always worried, and whenever the candidates will be selected, will there be such a extra step for, for the selection and things like that? But sounds like that shouldn't be. That's great. Thank you.
Thank you. Okay, we're going to move to Abigail's asked a number of questions in one, so maybe I'll break it down. So theoretically, what kind of selection process would there be for potential candidates, and would it primarily be for little ones rather than teens and adults?
So I think that is a challenging question, and I think the reality is that I just see that there being multiple programs for Rett syndrome, and I suspect that different companies may take different approaches, because there's no right and a wrong to this. Because the animal studies predict that you should see some sort of beneficial effect, whether you treat early in the disorder or late. In other words, whether you treat young children or adults or juveniles or young adults. But it's really going to be, so the conventional way for drug discovery and testing is that you test in adults first, and then if you and then you move into a pediatric population. But that's assuming that adults can make their own decision, and that's not going to be the case in Rett syndrome. It's going to be the parents or carers that are likely to consent. So there's no informed consent on the individual. So it blurs the line between treating adults versus under 18s. And then some companies, so that then there's the question, if you intervene early, is it likely that you'll see a bigger effect? And you could envisage that some companies will say, if you intervene earlier, you might be able to see whether it's having an effect or not quicker. And then other companies might say, or other trial designs might say, well, if you go in a later population, adult population, it may be that the clinical signs are more stable, And therefore, if we see an improvement over a stable baseline, we might be able to see whether we're having an effect or not more quickly. So I think there's lots of scenarios, and I don't think there's a right or a wrong. And I suspect that different companies will take different strategies here. So it's quite a hard question to answer that, really.
Yeah, so that's really a question of waiting for the protocols of the particular trial and the particular company. I guess the follow-up from Abigail there is also, would it matter which mutation type an individual has? Would those with mutation types considered to be more severe be deselected?
I don't think so. I don't see any reason, because I don't see any reason, because essentially in the animal studies, the gene therapies are tested in the most severe mutations. So they're the ones where they have evidence that the gene therapy is having an impact that's sort of driving the movement towards a clinical trial. So I don't think having a severe mutation is at all going to limit enrollment. It may actually be the other way around. It may be that if there's an individual that has a much milder form, the risk benefit is somewhat different. And but I don't really know. I'm sort of extending into areas of sort of clinical development, and I think a clinical colleague would be better to sort of comment on these things. But my feeling is that it shouldn't, any loss of MECP2 function mutation should be considered in these trials, I would imagine.
Okay, thank you. I'm imagining that's positive news for lots of people. Abigail, I don't know if you're able to unmute, but did you want to follow up?
Great, thank you very much.
Perfect, thank you. We've got a couple of questions along the same lines. So are the approaches, Taysha versus Novartis, significantly different? Is there reason to be optimistic that the Taysha therapy will be more effective than the Novartis therapy might have been? And a similar question is, several companies are working on gene therapy. How different are their products, if they are different? If that's...
Yeah, so, I don't want to comment too much on Taysha, because they're in terms of the difference between Taysha and Novartis, the main thing is that Taysha claimed to have a slight modification of the way that they do the gene therapy that prevents or reduces the likelihood of producing too much MECP2 protein. So that's their sort of selling point. Whether it works, I don't think we'll know until we reach a clinical trial. It's possible that their mechanism actually dampens it too much and they don't get enough. We don't really know. But yeah, there is a difference between those two programs in that sense. The actual where I would say that Taysha is unusual in that they're not restoring normal MECP2. They're actually delivering a very synthetic form of MECP2 that's much smaller than the normal protein, and they're assuming that it has normal function. So that's a difference between what they're doing and what other companies are doing. They have their data and they feel comfortable moving forward with that, but that's a difference. I think in terms of what other companies are doing, other companies are trying to look at other ways of sort of constraining the level of MECP2 to normal levels. And then lastly, and it sort of interleaves with the last question as well, there are some companies that are looking at different types of genetic therapy which target specific mutations. So there are programs in what's called gene editing therapy, which are treatments that are designed to fix specific types of mutation. So none of those have none of them have really presented the data publicly, but a lot of them are working on this. And I guess the difference with those approaches is that they should be highly effective, or they've the potential to be very effective, but they're going to be very effective for a specific. So it will be a drug that's developed for a specific mutation type. So I can't really say much about that because not very little of it's in the public domain. There's been some early papers published, but a lot of the companies that were on that second last slide are working in that area.
Yeah, thank you. I think we understand there's lots of confidentiality involved, and I think Rachel wants to jump in.
I just was going to say a question has just been added to the chat, and it's really not another question. It's just clarifying what you've already said, and it's about whether or not the gene therapy would be good for all MECP2 mutations. And the answer from what you've said is yes.
Yes.
Right?
Yes. Yeah.
Yeah. So it's more, I think, thinking about eligibility for clinical trials. It's more probably about whether or not somebody, rather than if they have. I think if they have the MECP2 mutation, they'd be likely to be a candidate at this point with the sort of clinical trials that look like they're coming. But they also need to have classic Rett syndrome rather than atypical, is what we're starting to learn, and that would be atypical/mild.
Yeah, I suspect so. I don't really know, and it's going to really depend on what the agreement is with the regulators, but I would imagine that the inclusion criteria would be 1, meeting the clinical diagnosis, and 2, having a confirmed MECP2 mutation are the sort of key things. And then I think what the likelihood is that there will then be other inclusion criteria like an age range, and there may be things like, I don't know, hand use or so. I don't actually know. I think it really will depend on the trial. I mean, I guess Tasha will present, they will announce what their inclusion criteria is ultimately.
Just to add in, there's another comment that's been made by Robert, who shared about his daughter being on the Anavex trial, and I think it's helpful to just clarify for a minute that what we're talking about here is, with gene therapy, is something that addresses the underlying cause of Rett syndrome. And so drug treatments like Anavex don't do that. They are looking at, whatever reason that the companies have had for developing that particular drug. It's looking at helping with symptoms, rather than going underneath and fixing the overall problem. Stuart, I might not be explaining that very well. Perhaps you can clarify.
Yeah, that's correct. I mean, the small molecule drug, the conventional drugs that are being tested are really treating some of the symptoms, whereas gene therapy is really sort of targeting the underlying cause of the disease. And because the underlying cause results in quite a lot of different clinical effects, it affects hand use and communication, and all of the things that you see across the spectrum of clinical features of Rett syndrome. It's affecting all of those, whereas the small molecules tend to be targeting specific aspects of this. So I think that's really the difference. But it may ultimately be that if there's a gene therapy down the line, there's no reason why there can't be combination therapies. I mean, combination therapies are very common, and it may be even with the gene therapy, there may still be use of combination therapy. So I don't view it as either one or the other.
Definitely not, because at the moment there are 0 approved drugs specifically for Rett syndrome anywhere in the world, and so it's obviously really important that we try things that that might help even in the shorter term or in the longer term for people who are not eligible for gene therapy. I think that might bring us to another question. Now, one of the questions was about whether or not the candidates would be, I think we already covered that one about whether or not the candidates would be adults or children, but I had a particular question about it as well. You were saying that in the adult trial, which in the Taysha trial, the adult trial is what's been announced. Basically, there's going to be a trial in adults in Canada. So what we know is that from what you've been saying, they'll do the safety testing first. And really that's the goal with the adults, is to just make sure it's safe, right? And so what I was wondering was, for those first few patients that are, and families that are brave enough to go for that, based on, I'm trying to ask the question in a delicate way, but based on the sort of existing literature and the mouse work that's been done, what kind of improvements might you see at the lower level of production of MECP2 protein that's likely to induce?
Yeah, I think it's really hard to say. It may be that so in the mice, when you intervene late when you switch the gene back on, you get correction even in adult mice. And I think there are probably, we don't have data on this, but my expectation is that certain functions might correct more readily than others. So for instance, I think things like what are called autonomic disturbances, so things like breathing, I think could correct, but other things such as when you get the rigidity or scoliosis and things coming in, and you've got a sort of tightening of muscle and things, that might be, there might be sort of physical things there that are then make it more resistant to correction if it's in the later stages of the disease. But I don't think we'll actually know until the studies are underway. And same with things like communication, we don't really know. That's something you can't really test in experiments in models and mice and things like that.
Yeah, I think rather than whether or not, in general, we hope to see an improvement from gene therapy, I suppose what I'm wondering is, because they'll start off with the really lowest dose possible just to show the safety rather than worrying about efficacy at that point, are those people likely to see any benefit based on what you've already, what's already out there? I mean, is it that if you, I think there's experiments that's been done, has there not, that show that you can get like a 30% improvement in MECP2 protein in the brain, or whatever different percentages, and what do those show? Because I just can't quite remember that.
Yeah, so certainly, with a mouse, so in the experiments, you don't need to put back 100% of protein. So a little protein is much better than having none at all. But that's at a single cell level. But when you look across the brain, what we know is that gene therapy, at the moment, the viruses that are being used are not efficient enough to deliver the gene therapy to every cell across the nervous system. It's actually a pretty small fraction of the cells. So that's what's limiting at the moment. But what I would imagine in the initial trials that will start is that they will look for measures that they can quantify and measure very reliably so that they can detect whether they're seeing signs of improvement. Because even going into, you know, adults with Rett syndrome, they will be looking for measures that it's actually doing, you know, having some benefit. I mean, I suspect that the reason that they're going into the adults first is that, you know, that was a sort of regulatory path that they took so that they could actually get into patients as quickly as possible. And once they've got safety in a number of adults, they can then move into a pediatric population as well. And I know that Taysha have sort of announced that they have plans also to look in males with Rett syndrome as well.
Thank you, that's very helpful. I'm sorry to ask a question. I think we all, as families, want the answer to questions that nobody can answer at this stage, but you do have that greater insight than the average person. So I appreciate you sharing those thoughts with us.
We've still got a few more questions in the chat. I'm aware we're a little bit over time, but if we could maybe just get a couple of these questions off. I imagine this is another slightly impossible question to answer, Stuart, I'm afraid. A bit more pragmatic though. What is the cost per person and who covers that?
Yeah, so for the gene therapy trials itself, then obviously there's no cost to the individuals because the sponsor of the trial covered the cost. And for gene therapy trials, normally, you know, if it's in a center, they would accommodate the family and the patient and cover those costs. For a licensed gene therapy, I think that's really going to be a question. So, you know, obviously Zolgensma is a bit infamous in a way in that it's this hugely expensive drug. And, you know, Novartis have been really criticized because of that, because it's quite so expensive. I think the real answer will be how effective the drug is will dictate how expensive it is. So if it, you know, if you have something that's, you know, producing a very profound benefit, then I think the companies have ability to charge more money for that. If it's a more modest effect, then it would be less. But I think that's going to vary a lot from country to country. And obviously, you know, the biggest market in pharmaceuticals is the United States, and it's such a different system to what we have here. You know, here people do get prescribed drugs for spinal muscular atrophy, which are very expensive. But you know, they get that because they're very effective. So I think that's what the calculation is going to be based on. But you know, as you're aware, the cost of not treating Rett syndrome is extremely high. So that will be taken into the calculation. But you know, that again, I'm happy to sort of give my opinion, but this is really a sort of question for the sort of people that work out how the NHS and others spend their money.
Yeah, I think we appreciate it's a very grey area. I don't know also if either Rachel or Andy just want to jump in and comment on just going back to what you said, Stuart, about being involved in the trial itself, whether either of you want to just say a few words about how we would support families participating in trials.
So
financially.
Typically, what's happened, I can only speak to what's happened with the UK clinical trials. We've worked quite closely with the pharmaceutical company to support recruitment of patients, but also then to help on behalf of the sponsor to work with families to arrange travel and accommodation. And so we've done it on the clinical trials that have happened so far, and the sponsor pays for that, not Reverse Rett. But we've so far what we've done is we've worked on a very individual basis with families to make these kinds of arrangements on the basis of what's happening in that particular study and the family's individual needs. And so I imagine that if we were to be involved in any proposed gene therapy trials that ever took place in the UK, we would have a similar sort of arrangement. The main thing to be aware of is that as much as humanly possible, the burden is reduced for the families taking part. So the idea is that the trial is taking part in a clinical trial, whether it's a drug trial or this kind of trial, is obviously stressful. It's worrying, it elevates your hope. It also makes you wonder, if you're doing the right thing, and questioning yourself and the responsibility you're taking for that person's participation. And so we don't want people to have to be worrying about having to get the train or where they're going to stay and if it is accessible, etc. So that's what we do there.
Yeah. So I guess not sure whether the question really meant what is the cost once it's approved, which is unknown, but in terms of the cost of actually being on a clinical trial, there shouldn't be any financial burden to a family. And another hopefully practical question, how long does it usually take from when gene therapy starts in clinical trials versus when it reaches the market? Is that answerable?
Yeah, there's not a lot of precedence for this because there's so few gene therapies reach the market, but I think it really depends on the disorder as well. For instance, there have been gene therapy trials for haemophilia that have been going on for a decade or more, but they've never reached licensed drugs because there are other existing treatments out there, and therefore the bar to be safe and more effective is much higher. But for something where there's huge unmet clinical need and there's no existing treatments, there are potential to accelerate, but they really need to demonstrate proof of concept that it's actually working and is in benefit, and to get the label on the drug that it's approved in a particular section of patients, they need to have tested that patient. So it may be that they broaden the, so if they have tight inclusion criteria, there may be a follow-up trial with broader inclusion criteria so that they can get a broader label. So it really just depends. But Zolgensma was quite accelerated, and compared to traditional drug discovery is very, very slow. Gene therapy tends to be a bit faster, but then all of the early gene therapies have been in very severe disorders which have facilitated that. But Rett syndrome would fit in that. But I wouldn't like to put my head on a block and say X years, because I don't know what that number would look like.
Just to chip into that one, Stuart, I know that when you were doing your explanation of how the gene therapy products are made and the incubation process and everything else, I think it's been the case with Zolgensma that they hadn't yet done a great deal of manufacturing before they started the clinical trial. So there was a bit of a delay there, but I think it was altogether has been about 5 or 6 years.
Yeah.
But I know whenever we spoke to people from Avexis in the kind of earlier stages, they did stress that they'd had these delays in manufacturing, which hopefully other companies have learned from and are prepared for. The thing that I do think is going to take the time as well, though, is making, this going through the regulators and the payers in the UK and in Europe. It's not going to be a question of it just goes through and everybody has access immediately. It's going to take some time.
Yeah.
And that's just the way that the system is here. But we are doing everything we can to prepare for the inevitable, I don't want to say battle, I will say process of engaging with the payers. And when we get to that point, it will be a great problem to have.
Okay, I think we're moving slightly back to science rather than the pragmatic questions. Is there evidence of the brain growing in mice models? And if so, what are the implications of that in human therapies?
Yeah, I mean, that's a very good question. So yeah, so in the mouse models of Rett syndrome, the brain is smaller, and we know when we intervene with genetic treatments that we get increased growth. You know what that means clinically, we're not, you know, we don't really know, because obviously the skull is of a finite size, so you can't really grow the brain because it's constrained. But I think it really probably depends on the age of intervention. But you know, there's evidence for in mice that the complexity of the nerve cells increases. So in other words, they form more connections and they have more branches, the nerve cells. So we can actually see that taking place when we switch the gene back on in mice, and that has been published. Yeah, of course, these are things that you can't measure clinically, but you know, there is an expectation that so it might not be physical growth, but the actual connections may actually grow and change. And you know, that's termed synaptic plasticity, and that is the synapses are the connections between the nerve cells. And that again has been shown in mice, that when you put back MECP2, then you get a lot of these changes occurring, which occur at the same time as you see improvements in the sort of clinical features.
Yeah, I think Abigail's got a follow-up, quite a similar but follow-up question in a way. She says, what happens if the brain expands and forms new neural pathways, as you were just explaining, as a result of successful gene therapy? Could this put increased pressure on the skull in individuals whose skull plates have fused, e.g. older than adults? Is that a possible side effect?
It's a theoretical side effect, but I think I mean, there's a lot of safety studies go on before a drug is allowed to go into a patient. And you know, for instance, you know, Taysha have, you know, announced that they have conducted studies in large animal that they've given their therapy to, and that's been safe and well tolerated. So I think it's a conceptual risk. So this came up. There was a drug for the so there are what are called trophic factor drugs, the tripeptide, the IGF-1 tripeptide. You'll remember the name for that. But you know, that is a growth factor that causes the brain to grow. And I know that in those clinical trials, they did measure intracranial pressure as part of the clinical trial to actually assess whether there was any increased pressure building up in the cranium.
Thank you, Abigail. Does that answer your question again? Just coming back to you. She might not be listening. We'll move on. We've got a couple more questions in the chat box, which we'll address, and then I think any further questions that come in after that, we might pop in an email to Stuart for him to respond to in his own time. So we talked about gene therapy theoretically being fine for any mutation. Kurt has asked, is there a difference in gene therapy for a mutation or a deletion? Would there be any difference?
No, I don't think so, because basically you're putting back functional protein. So whether you've got MECP2 already there, but it just doesn't work, or whether you have no MECP2, you're kind of just bypassing that and putting back functional protein. So we've actually modeled this in animal models. So we've looked in models that either lack MECP2 or models that have MECP2 that doesn't work, and we see a therapeutic effect in both. So there's no reason to suspect that there should be a difference between those two scenarios.
Okay, That's excellent news. And possibly putting you on the spot a bit again, Stuart, with a slightly unanswerable question, but do you think there will be a clinical trial in Europe before Canada's results are published?
I would say I doubt it because they're starting quite soon. So I think that is very unlikely. You know, as I say, there are various companies that are working away quietly, but I would be surprised, pleasantly surprised, but I don't think that's likely to be the case, because, you know, it takes time to, you know, get approval, and then even once you've got approval, you know, putting in place the clinical site and all the safety committees and things. I just think it's probably unlikely. But, you know, what may happen is that, you know, yeah, I don't think within a year, no, because they've announced that they hope to have an initial announcement of early findings by the end of the year. So I think it's unlikely, but
as you say, and I'm a happy surprise if that's different, and we won't hold you to anything on. Yeah,
I mean, there may be other announcements for, you know, plans for trials and things, but I just can't imagine one starting. But it would be nice. And then, of course, you know, whatever company, you know, if there's encouraging data from a trial that's opened up in one sort of geographical zone, then, you know, that's impetus then to open up more trial sites. So it may be that, you know, encouraging data from the Canada study would sort of trigger opening of trial sites elsewhere, you know, reaching out to the UK and European and other regulators.
Okay, I think, as I said, I think we'll stop with the questions there. If anyone does have any further pressing questions, feel free to email them to me, and I can then pass them on over to Stuart, and I'm sure he'll be willing to hop on. I think, and thank you. Excited at the moment really help in layman's terms, as Rachel said, done a fantastic job of putting that in language that we can all understand and clearing up a lot of the questions that I think we have. So thank you so much, Stuart, for sharing that all with us. Thank you everybody for joining. You will receive a link to the recording of the session afterwards. So if there's anything you missed, or if you want to go back and have another look, and also obviously you can feel free to share that link with others. It will be on our YouTube channel, and you can find all our previous Bitesize sessions on there as well. So anything you miss, you can go back to. I'll pop back over to Rachel to lend her thanks and close as well.
Okay, I just would like to add, you know, to Beth's appreciation to Stuart. It's been a really interesting session. And I know there's been a lot of questions about people's expectations for what's going to happen next. And that just speaks to the excitement and passion that we all have to see this work come to fruition. I think it would be really encouraging to tell people that if you weren't on the recent FDA drug development meeting about Rett syndrome, which was hosted by IRSF and RSRT, when the person from the FDA spoke at that meeting, he did say, you know, we are looking to get gene therapies through approval as fast as we can. If we start to see clinical trials looking positive, we're not going to ask companies to go to bigger clinical trials. We're going to just try and bring these treatments forward. But we are at, as Stuart has said a few times, really, we're in a new era here of really kind of life-changing treatments for people, and a lot of this hasn't been done before, or it's been done in slightly different circumstances, as with Zolgensma. So some of it is going to be we wait and see. And as always at Reverse Rett, we remain as optimistic as possible, and we're always pushing for things to happen more quickly. We know that all these children, young people and adults with Rett syndrome are waiting. And we remember that every day in the work we do, as I know they do over at University of Edinburgh in Stuart's lab. So Stuart, thank you once again. Thank you to everybody for coming today, and hopefully see you again soon.
Thank you, everyone.
Thank you, Beth. Thanks, Rachel.
Thank you.
Bye.